Okay, so welcome everybody to our Smart Science Journal Club. We're really excited that you are here with us today, and we have some really great speakers um, and some really great talks. So we're just really excited to get started. I'm going to ask our first speaker to come on, and I'm going to introduce him to you. So today's first speaker is John Lovell, and he came to genomics by way of ecology and plant physiology with an interest in what processes govern plant distributions in nature and productivity in agriculture. John received his PhD in ecology from Colorado State University, where he studied genetics of drought adaptation, focusing on Arabidopsis and its wild relative Buchera. He then did a postdoc with the DFG Fellowship at the Leibniz Institute of Plant Genomics and Crop Plant Research, and then a second postdoc at the University of Texas with the NSF Fellowship studying genetic basis of drought adaptation and stress tolerance in panicum grasses. Since 2017, he has been at HudsonAlpha integrating comparative genomics with quantitative genetics and PopGen experimental design. He works mostly with plants, and his active projects include biofuel feedstocks, mosses, ferns, forest trees, wildflowers, orphan crops, and cash crops. So please welcome John Lovell.
Thank you, Nikki. Thanks everyone for joining today. So a little bit more, I'm based at HudsonAlpha Institute for Biotechnology Genome Sequencing Center in Huntsville, Alabama. I actually live in Denver, Colorado, kind of right in between the two major places that we do business. The HudsonAlpha Institute for Biotechnology Genome Sequencing Center, we sequence, assemble, analyze plant genomes, and a lot of our work is done in collaboration with the Joint Genome Institute, which is a Department of Energy user facility in Berkeley, California. I have affiliate position there. And in addition to doing DOE mission-based research with the Joint Genome Institute, we also, the Genome Sequencing Center at HudsonAlpha, we also do a lot of, a variety of other projects unrelated to DOE, including ecological and restoration genomics. And what I'll talk about more today, food crop development and genomics, including Pecan. So our major goals in our group are really to better represent the genetics and genomes of our study organisms. The leaders of our group, Jane and Jeremy. Jane does the wet lab and molecular stuff, and Jeremy leads the computational side. And I lead the evolutionary analysis group under Jeremy. And so in addition to actually generating the genomes and other resources associated with genomes, we do a number of computational approaches to try and better connect our genomes that we produce to biological questions. One way that we do this is through comparative genomics. I'm showing below here a map of synteny among a number of mammal genomes from NCBI. Ultimately, our goal is to then leverage our genomes and population genetic and quantitative genetic information to make meaningful impacts and improve agricultural productivity, for example, or environmental sustainability. One example of this comes from our work recently in switchgrass, where we built a single haploid representation of both subgenomes of the tetraploid switchgrass genome, aligned reads from several hundred genotypes collected across the eastern 2/3 of the United States, and combine that information with common garden phenotype and biomass yield data. And what we're able to find is that there are a number of regions in the genome that have evolved an upland ecotype, which is susceptible to heat and pests, but very tolerant to cold and drought. And that these regions of conversion from lowland cold susceptible genetic background to an upland cold tolerant background likely permitted faster adaptation to the northern habitats in North America following glaciation. But in this analysis, which I think is really neat, we used just one haploid representation of the genome, essentially a single haplotype, but we know switchgrass is outbred and there's a tremendous amount of diversity within switchgrass. Really what we want to do and what we're going to be doing in the very near future is building out genomes that represent both meiotically homologous haplotypes in the genome plus genetic diversity and represent the genetic diversity across this very diverse species. In this effort to better represent the genetic diversity of a species in our genomes is not just a goal of ours, but a goal of, I think, the whole field of genomics, and is well underway. Recently, genomes across 26 maize genotypes that are the name founders came out, as well as nearly gapless complete genome assemblies for multiple haplotypes in Drosophila, outbred genomes in potatoes, and many, many other systems. So the field is already starting to make genomes that better represent the biology of the organisms that we're interested in, which is really a big step. So I'll talk today about our efforts to do just this in Pecan. This work came out in Nature Communications earlier this year, and the resources that I'll talk about today were the assemblies were built by Jerry and Chris, our group, and Avinash did the annotations with help from our colleagues at Joint Genome Institute. What we did is we built 4 reference genomes. 3 of these are using the older PacBio consensus long read technology and have fairly reasonable contiguity. Let me see if I can use my pointer here. Yep. So you can see that these 3 genomes, the Oaxaca, Lakota, and Elliott genomes, pictured, these are the plants here that were sequenced, have contig N50s, so half of the contigs are greater than 3.5 to 5 megabases. You can see that information here, and they capture the vast majority of the sequence. But Pecan is, maybe for those of you who work in like inbred crops, for example, the genetics and biology of Pecan are actually more similar to you than your study organism. In Pecan, there's more genetic diversity within an individual than between any 2 individuals in a population. There's a tremendous amount of heterozygosity. And so in pecan breeding, since these trees take a really long time to go from one seed to another, a lot of the effort is done within a sibling family. The same reason that you are different from your brothers and sisters in terms of your appearance is the same reason that any set of seeds that you pull off a single pecan tree will produce very different offspring. And so it's that level of selection among siblings, that's often what leads to improvement in pecan. And that is crucial for us coming from building a genome perspective, because we want to be able to provide the breeders with the most accurate representation and most useful representation possible. And if the breeders are really all trying to combine information and look at the divergence between 2 haplotypes within an individual, we should at least be trying to build a genome that has both outbred haplotypes in it. And so that's what we did for the 3 CLR genomes. Our alternative haplotypes were pretty good, but could have been better. You can see that they cover between 65 and 75% of the sequence in the primary. And importantly, they're also broken up into pretty small pieces, contig N50 of 100 kb. This is due in part to the biology of these individuals, in that there is some inbreeding in the history in the pedigrees of these plants. So we get runs of homozygosity, and if you have smaller reads or less accurate reads, these can lead to collapsing of the 2 haplotypes. So you go from a region of 2 haplotypes down to a region of 1 and back to it. So that's why you get the smaller haplotypes and the shorter and the less coverage of the whole genome. In contrast to those 3 CLR genomes, we built a PacBio Hi-Fi CCS genome for Pawnee, this cultivar right here. Its primary path contains 100% of the expected sequence and has contig N50 roughly 6 times the size of the other genomes. Importantly, the alternative haplotype, the path through the genome that represents the meiotically homologous region to the primary path, is almost as contiguous as the primary path of the CLR genomes. About almost 3 megabases contig and 50, capturing almost 90% of the sequence. We think this is fairly accurate, a fairly complete depiction of the alternative haplotype for Pawnee, primarily because there are these runs of homozygosity. So we don't expect to get 100% of the sequence, and we could never get contigs that span, for example, the whole chromosome, because we'll hit regions of homozygosity. So now we have 4 outbred genomes, essentially 8 genomes. And this is great because we've accurately represented the genetic diversity of Pecan, but what do? All of the programs and computational framework that we have really relies on mapping short reads to a single haploid representation of a genome. So we needed to reconceive how we want to analyze these genomes in terms of combining quantitative genetics into our genomics. And it turns out that we already have some of the framework to do this. If we think about just, for example, in Pawnee or just within the Lakota genome, how can we analyze these 2 haplotypes? It's essentially identical to analyzing 2 haploid genomes sampled from a single population. And we've already built the framework for this. A few years ago, we built the genomes for a lowland and an upland inbred diploid relative of switchgrass called Panicum hallei. The 2 genomes are the HAL2 and the FIL2 genomes, the representatives you can see over here. And these 2 genomes, they're haploid representations of inbred individuals. So they're actually an accurate representation of the genetic diversity within those individuals because they're inbred. Since those 2 haplotypes are sampled from a population that should theoretically recombine and shares an evolutionary history, is essentially identical to building a outbred genome. So what we did with the HAL2 and FIL2, what we were interested in is understanding the evolution of drought tolerance, where the HAL2 genotype is really drought tolerant but doesn't grow very fast, and the FIL2 genotype is very drought susceptible but grows quickly. So we were trying to understand the trade-off. We built a genetic map and a eQTL experiment, or a quantitative trait experiment looking at gene expression between these 2 genotypes. And we found a couple quantitative traits, loci, regions of the genome that causally affect variation in a trait, one of which was on the top of chromosome 3. And since these 2 genomes we knew were completely collinear or syntenic, that's what these pale blue regions or polygons are. If we take a QTL interval, we can just mask the rest of the genome outside of this syntenic region, ask what genes are in there, and find ones that are particularly interesting. In this case, the likely causal gene is an abscisic acid overly sensitive gene, that Werke transcription factor that likely affects responses to drought. All right, so we have this framework here to compare 2 genomes, but we wanted to make this generalizable across multiple genomes. And to do this, for the last several years, we've been working on a pipeline called Genespace. Genespace was released recently as an R package, and you can get it on the JGI GitLab repo, pasted here, or reach out to me and I'll direct you to it. Basically, what Genespace does is it takes any 2 or more whole genome sequences and constrains blast hits between those 2 sequences to synteny, to known regions in the genome. And synteny can be perfect, like what we saw with Panicum hallei, or it can be highly fragmented. Either way, once you constrain to synteny, to known graph edges between 2 genomes, and you know that there's this one region that is syntenic, once you've constrained to that, you can pull out orthologs much more effectively. Here, for example, is what the syntenic graph network looks like among a few mammal genomes. But importantly, for quantitative trait exploration, and especially in between multiple genomes and trying to figure out what candidate genes might underlie a QTL, The big thing that this framework allows us to do is to find a region of interest and track the position of that region of interest across multiple genomes. Here, for example, if we had a QTL on the right arm of chromosome 5 in Panicum hallei, which is a model for plant growth and stress, we could say, okay, there's this variable region in Panicum hallei. What does that region look like in, for example, sorghum or rice, or the genetic model for grasses, Brachypodium? So we can constrain to just a synthetic region and look for orthologs therein. All right, so we applied this framework to Pecan, and one of the reasons that it's really important to do this in Pecan is because Pecan has a fairly ancient whole genome duplication that we want to functionally ignore. I'll just say that if we look at dot plots, chromosome 1 and 2, for example, are paralogous, and these hits off diagonal, those are, we don't want to explore those in terms of our PanGenome framework. So we go from a network that looks like this, that has paralogous matches between chromosome 1 and chromosome 2, between our 4 Pecan genotypes. We mask those and we end up with a single copy, clean synthetic representation across our 4 Pecan genomes and to the outgroup walnut. With the synteny constrained framework in place, we can then build out a pan-genome annotation, essentially asking for any particular region in the genome, what are the genes that are orthologous in that particular region between Pawnee, Oaxaca, Lakota, Elliott, and the outgroup. And this lets us not only ask about protein similarity, but also a presence-absence variation. So for example, here there are about 20,000 genes that are present in all 5 of these species. So now we get a database that is projected against the physical positions of any of these genomes that has not only protein similarity in it, but also presence-absence variation in the genes. And we can use that and query, for example, a quantitative trait locus interval to ask about potential candidates. So we did just this using the Lakota genome. So Lakota, like I explained before, like most pecans, is essentially an F1 between 2 diverged parents. In this case, blue is coding the pollen donor, Mahan, and yellow is coding the maternal parent, Major. And we produce an F1 population with some outgroup, in this case, Oaxaca, that is a random distribution of the grand parental alleles. And then we can ask, are there regions in the genome that are associated with, that associate a trait with variation between the 2 haplotypes in Lakota? In this case, the trait is pest resistance. These are galls formed by phylloxera, which is an aphid-like insect that really can destroy the leaves. And this trait spans anywhere from almost complete destruction of the leaves to no impact at all in these individuals. And here I've plotted the haplotypes of Mahan and Major, the grand parental alleles in this F1 population across chromosome 16. We can see there's a massive QTL here. That's what this peak represents with a LOD score on the y-axis. It's 1.2 megabases, highlighted by these 2 white lines here. And you can see that pretty much all of the susceptible individuals in this population have the yellow allele, and most of the blue individuals are completely resistant. So we can query the PanGenome in this 1.2 megabase region. And this is what the PanGenome looks like, just comparing the primary and alternative haplotypes in the Lakota genome. This is the primary path here. It's almost all the major allele, which is the susceptible allele. You can see first that there's a large gap here. This large gap represents a region that we think is completely homozygous, so we don't get an alternative haplotype in the region. Each of the points here, a filled circle, represents a 1-to-1 ortholog between the primary and alternative haplotype. If there's a thick bar, that means that those proteins are pretty much identical. All of the open circles up here, those represent presence-absence variation. So the thing to notice here is that there's this large run of genes that are only present in the alternative haplotype They're not present in the primary haplotype of the genome assembly. So if this was a haploid genome assembly that we're putting out there, these genes would not be present. Yet this is part of a large run of genes that are annotated for disease associations in other species, and particularly leucine-rich repeats that we know might underscore tolerance and susceptibility to things like phylloxera galls. So in total, there's about 20 genes here that are highly enriched just within the resistant haplotype and are not present in the primary, that likely drive resistance to this pest, and show how important it is to represent both haplotypes when doing anything with outbred species. So with that, I'll just say that pretty much now, especially with hi-fi assemblies, we can build outbred genomes for maybe the majority of complex plants, at least as long as they themselves are heterozygous. It's really great. And that as we move forward, we're going to be seeing multiple de novo genomes within most species, which is going to require a comparative genomics framework to connect variation among these genomes. And lastly, that while this is really exciting that doing this PanGenome annotation framework allows for this, connecting variants like from short reads like SNPs, indels, that are not present in the whole genomes, that is a difficult challenge and one that we're still working on. So with that, I would just like to thank the team. Our colleagues working on Pecan include folks at New Mexico State, University of Georgia. Sequencing was done by Jane and her group, and was funded by the USDA Specialty Crop Research Initiative. And with that, just lastly, I'll just put out a quick plug to say that working with the Joint Genome Institute is really great, and if you have the opportunity to do it, I highly recommend it. We have community science program calls out pretty much continuously, including large-scale calls to advance the genomic resources of your study system, as well as smaller new investigator calls. All right, thank you.
Thanks, John. That was really great, really fascinating, and I think you're going to have a pretty active question Q&A session after this. So thank you very much.
Hey, John.
Hey there.
Good job. That was a really good talk. They were both really good. All right, let's see. Okay, well, I'll go ahead and fire away the first question. John, it's kind of a long one, like a two-parter. So if you want me to repeat it, let me know, or I can repeat just the second part. Okay, so when thinking about representing diploid or polyploid genomes, have you considered including the runs of homozygosity in both the primary and alternative haplotype sets to give a holistic view and enable analysis across two haploid whole genomes? Building on that, if you are masking out the homologous chromosomes from the comparisons amongst the four genomes, is there value in generating a phased genome?
All right, I'm going to answer the first one first, and then we'll come back and swing around to that one. So the question of representing sequence that is identical between two, let's say, haploid or two meiotic haplotypes, meiotically homologous haplotypes in a single genome is an open one. I think the best analogy to that might be like the human X chromosome, and that compared to the Y chromosome, there's the sex determining region that is completely distinct. And so if you'd want to represent both of those uniquely in your assembly, but there's also the pseudo-autosomal region that can be identical between the two. So when you're building out a genome assembly, in the case of the human X and Y, you want to present both, because the run of homozygosity and the region that's completely identical between the two is very small, just 100 kb or something like that compared to the whole genome. But if you compare, let's say, two sex chromosomes in asparagus or a plant, the region that's identical is, I mean, the region that's different, that segregates between the two, is tiny, and the region that's identical is gigantic. So do you want to represent those as two separate chromosomes in a quote unquote haploid assembly? It's one that we don't have a good answer for. And I think certain situations you do want to represent those runs of homozygosity, and in others you don't. And for sure, in places like in the example I used in Pecan, where there's that region that we can't assemble, we do not want to put that sequence into both assemblies, because we can't be certain that it's there versus that it's just collapsed. I think maybe in the future, now that we're doing high-C informed diploid assemblies, so that the high-C is actually used when building out the two genomes, I think that now we have the chance to maybe better phase that region and produce that the identical sequences, but that, at least in the technology that we used to assemble in Pecan, that wouldn't have been feasible, I don't think. All right, phasing in the masking prologous regions. Maybe whoever asked that question could come on and explain.
Yes, I can do that. Okay, Michelle, are you there?
I am.
Yay.
Sorry, it's a PacBio person asking these questions. I was just really interested. Yeah, so they were kind of two separate questions. Thank you for that first answer. That's super helpful. I agree that unless you can be 100% sure, because my mind initially goes, yeah, but there is, like, if it's truly identical, like there really are two copies of it though, right? So you can copy it. But like you said, if you can't be 100% sure, it's not worth throwing it in there and being wrong. So that makes complete sense. Hopefully we'll have better resolution in the future. My second question is, when you're using your tool to compare the different genomes and using those syntenic regions, and you're masking out kind of the second copy of each chromosome, then I'm wondering, like, is that a waste of generating a phased genome? Like, what added value does that bring? Or is it just generating the phased genome up front gives you a better representation of an accurate primary contig set?
So I think this is maybe something I didn't explain adequately. The reason that we are masking is not because we didn't want to have phases between our two assemblies in the diploid, but because this is a plant and plants have experienced a whole genome duplication, that there's actually, in this diploid, there's actually 4 copies of a lot of genes. And that 2 of those copies, while similar, they're paralogs, meaning that they arose from a duplication event, while they're similar and find their way into orthology graphs based on sequence similarity, they are not, how do you explain this? They're not informative if you want to look at a specific region of the genome for breeding. So chromosome 1, while similar to chromosome 2, at least at the protein coding level, chromosome 2 actually has nothing to do with chromosome 1 when you try and look at a QTL. And so you need to mask that. You need to break those graphs, those orthology graphs based on synteny, and remove these paralogs. Does that make sense?
Yeah, it does. Yeah. That helps. Thank you.
Okay, thanks, Michelle. Great questions. Okay, we have another hand up. So can you use Genespace to quality control genome assemblies and annotations?
Yeah, I left a bullet point off on the summary about that. Yeah, so if you expect that you have, let's say, a primary assembly in your alternative path, if you expect them to be, meiotically homologous and that there shouldn't be inversions in there, then when you compare those two in terms of synteny, if you see inversions, there are 2 options: one, that it's an assembly error, or 2, that your individual has a real inversion. You can look at the block breakpoints right around that and ask if they correspond to different contigs, and if so, maybe you need a scaffold differently. So yes, we can, and in fact, we're working on a pipeline right now to automate it, to do that in an automated and fast fashion, because it's pretty quick.
Okay, thank you. In the meantime, another question: how would the candidate gene search work in polyploids or inbreds instead of outbred diploids?
Yeah, that's a really good question. So it depends how the polyploid works. Let's assume that it's a disomic polyploid, and that you really, even though it is a polyploid, there really only are two possible outcomes at a specific region. In that case, you just would do it exactly the same way because it's disomic. If it's a true polysomic polyploid, meaning that all of the chromosomes that, if you have a tetraploid, all four of them can recombine, man, good luck. It's so hard to do QTL mapping and candidate gene searches that way because you can't track the haplotype that actually drives it. Often it's better to just visualize that as a monoploid path, as a single path, and then ask about the divergence of the alternative chromosomes against it. In terms of the inbred assembly, well, you need another assembly, right? There's no diversity in a haploid assembly. So if you want to compare, if you want to do a QTL analysis and candidate gene search within an inbred diploid or a haploid for that matter, you just need another genome like we did with Panicum hallia. So you compare two or more. And so that's how it would be. If you have short reads mapped to just a single reference, you don't need Genespace at all. You do your standard candidate gene search through SNP-F annotations or some other functional variant function annotation software.
Okay, thank you. So I'll read, we saw a question come through in the chat. From a QTL mapping perspective, how might the use of a pan-genome as a reference impact the development of a linkage map, especially in the presence of PAVs such as you presented in Pecan?
Yeah, great question. So you're talking about actually like building the map. All right, so let's think about it in terms of the maze NAM where we have 26 founders. And in that case, essentially what you can do is build out a map based on k-mers that are different between all of the parents, and all of the individual sequences that went in, and you can essentially build a haplotype graph out of it for each individual. And then QTL mapping, boom, you're just doing it straight on those k-mers against a specific reference or against a common coordinate system. Depends how you want to do it. The PAVs, it shouldn't matter because you should know in your genomes whether you think that those PAVs are real or induced by the assembly. In the case of Pecan, with that gap, with one of the gaps, that PAV, we can say that it's not real because we don't get an alternative assembly there. But if it was a hi-fi assembly, we might be able to, like what I said in the earlier question, we might be able to discover that. And then in the other one where we have coverage on both regions and just can't build gene models on one of them, that's really strong evidence of a true PAV. Either way, I don't think it would affect the downstream analysis in terms of candidate gene discovery and the development of a linkage map. You have to do a different approach than mapping to a single reference, or you map to a single reference and then propagate the positions of that mapping to a common coordinate system. Yeah, so I guess maybe that's not a satisfactory answer. I'm not sure, Josh, but if it is, if it's not, feel free to ping me on Twitter or whatever. I'm happy to talk about it more. All right. Oh, Dong-ha got his questions there.
Yes. I can go ahead and read it just for those who are listening. Have you tried using on more divergent genomes, for example, different closely related species in a family to catalog CNVs, etc.?
So I think you mean less divergent genomes if it's members of in a single family, or not. Well, either way, yeah, copy number variation is something that's spit directly out of this program. So you know the number, as long as they're, so let me step back, as long as the copy number variation is syntenic or a tandem array, for example. So as long as the copies have expanded within a syntenic region, you'll get all of those copies in there, and you can just count how many. And there's a routine that just tells you whether it's a single copy and how many copies there are for every gene. So you got a big matrix. If it's closely, if you mean like closely related genomes, as long as they're really good genomes, it should work exactly the same. Really distantly related genomes where synteny is not conserved, like comparing a platypus to a human or a Arabidopsis to cotton, really, really unpreserved synteny. A problem arises where if a tandem array occurs at a syntenic block breakpoint, you'll split the copy number variation into two specific regions. So you might have something that's truly a 5-to-1 expansion, but it's actually in the PanGenome framework, it's split into a 2 and a 3. And this happens, and it's just the only way you can go about it if you want to constrain to synteny. And if you're really interested in like true gene expansion, maybe you don't constrain to synteny, and Genespace also has that option.
Cool. Okay, well, I think that's all the questions. Unless there's any last minute questions, you can throw in the chat really quickly. If not, John, thank you so much. Wonderful talk, great conversation, and thanks everyone, and have a good one. Thank you again, John.
My pleasure. Thank you, Michelle. Take care.
Yep. See ya.